All right. Continuing our discussion here on the molecular structure of DNA and RNA, this is part 3. So we had walked through some of the work of Linus Pauling, of Rosalind Franklin, and Erwin Kargaff. So if you remember, this individual kind of solved some crystal structure patterns demonstrating a helix in proteins. Rosalind Franklin did some crystallography on DNA and came up with some key facts um, about the overall structure. And Kargaff worked out that there are equal numbers of A's and T's and G's and C's, right? So this really comes down to what Watson and Crick had to work with. So they had information from all those experiments, and what they did is essentially sat down with a ball and stick model of molecule or of atoms and started to try and build molecules that would fit all of the key parameters of previous experiments. Now, they had actually been doing this for a while and had come up with plausible structures that turned out not to be true. But with each new piece of information, they were able to work out the additional aspects of that. So one of the key things here is the sugar phosphate backbone is on the outside and the bases project in towards each other. And this was not the initial thinking because the bases are really the only interesting thing about DNA. So everyone was trying to put them on the outside. Now, when they were first starting to try and build this, they kept trying to match A's with other A's and T's to T's and C's to C's. And this makes logical sense of if you have two strands and they're copies of each other, this is the most logical way of working it out. But of course, this doesn't really work. It was when they started incorporating Kargaff's work and they realized that A's and T's were fitting together and G's and C's were fitting together, they were able to build their overall model. Now, ultimately, Watson, Crick, and Wilkins were awarded the Nobel Prize in 1962. Rosalind Franklin would have been included in this, but she had died in 1958. And unfortunately, Nobel Prizes are not awarded after death. So, as I said, there's more to the story of Rosalind Franklin and Wilkins and Watson and Crick than we have time for here. Maybe we can discuss it if that's something you're interested in. But let's go ahead and move on and look at the actual features of a double helix. So I want you to be able to outline the key structural features of a DNA double helix. I want you to be able to compare and contrast the B form of DNA with Z and A forms of DNA. This is less of interest. The B form is really what we want to concentrate on. And we can also take a look at something new that has been discovered, and that is a triple DNA helix. So, some of the general features. First of all, we have two strands that are twisted around a common axis. There are 10 bases per complete turn of the helix, and that spans about 3.4 nanometers. The two strands that are in this are anti-parallel. So while one runs 5' to 3', the other is running 3' to 5'. Okay. The helix is also what we call a right-handed helix. So what we mean by a right-handed helix is as a helix is spiraling away from you, the helix turns in a clockwise direction. Now you might ask, well, what does this have to do with right-handed versus left-handed? Well, this is better illustrated by this picture. Okay? If you take a look at this helix, it is progressing with rotation that is clockwise if it is moving away from you. This is moving counterclockwise as it moves away from you. What this has to do with right hands and left hands is if you take your right hand and pretend like you're holding a rod or cup in your hand so that your fingers are curled around, if you move your hand in the direction of your thumb and twist your hand in the direction that your fingers are pointing, you will move in this type of helical formation. Where with your left hand, since your fingers are pointing the opposite direction, you're still going up, but you are twisting in the opposite direction. So this is what we mean by right-handed versus left-handed. Again, looking at me doing it in class, demonstrating this will help you solidify that. All right, so the DNA double helix. So the things that really stabilize this overall structure is, number one, we have hydrogen bonding between complementary bases. So A's bind to T's with 2 hydrogen bonds, and C's bind to G with 3 hydrogen bonds. If you recall, all the nitrogenous bases are kind of flat shapes in structure. So the fact that they are flat means that you can kind of stack them up on top of each other. And these two facts together with the limitations on the different bond angles that naturally exist with carbon, you build this overall structure. So let's take a look at this double helix. Here we can see we have the 5' end, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And you can see that there are the bases facing inward. Okay, so here's 5' and 3' end of one strand. The other strand is starting off down here at 5', wrapping around, and now we have 3'. So they are anti-parallel. We have G's and C's matching up, and they have 3 hydrogen bonds. And we have A's and T's matching up with 2 hydrogen bonds. So here is one complete turn from here to here. So if you look at this spot across here, it's the same as over here. So this is one complete turn. If you were to count the bases here, you would see that there are about 10 base pairs. And that the length of this is 3.4 nanometers. Now, taking a look over here, we can get a more fine-tuned picture of how the base pairs hydrogen bond. So here we have an A, and on this A we have a nitrogen that has a double bond and another single bond to carbon. It has a lone base pair of a lone pair of electrons. That lone pair of electrons are going to attract this proton from this amino group. Up here, we have this amino group right here, and it has hydrogens hanging out. Remember, the nitrogen is a little bit more greedy than carbon, so it tends to hoard the electrons. Over here, we have the opposite scenario. We have an oxygen double bonded to carbon. It has 2 lone pairs, and it is very greedy for those electrons. So its lone pair electrons are going to attract a hydrogen from over here. You can see a similar scenario happening with G's and C's, but you can see that we now have 3 hydrogen bonds as opposed to just 2. And if you look here, it doesn't matter how you put this in order. The fact that we are pairing a purine with a pyrimidine means that the overall spacing between the two is the same. It doesn't matter if the purine is on the left or on the right. Since you have a pyrimidine counterpart to that, it always ends up with the same distance. Okay? Now, some other key features here is if you look at the DNA structure, there are a couple of grooves running along the outside of the molecule. There is a large groove, which we call the major groove, and then we have a smaller groove called the minor groove. Looking at it here, you can see that the strand right here and the strand right here, these are fairly close together, but then there's a big gap in here. So this big gap is called the major groove, and this little smaller gap is called the minor groove. A key important aspect of this is on the minor groove, you cannot physically reach in and touch the nitrogenous bases in the middle. Okay? The major groove, you can come in and make contact with them. Okay? So when proteins are binding DNA in a non-specific manner, that is just recognizing the backbone, it's generally binding along the backbones themselves and along this minor groove. When you have proteins that can bind to specific DNA sequences, it can do that because it is binding in this major groove, and it can actually see and usually hydrogen bond with these base pairs. Now, if you take a look at the structure here, the bases have kind of a flat-like appearance, and you can see that they literally stack on top of each other. This is something that gives a lot of the overall structure to the DNA, the fact that they can stack in this manner.